Hello folks, we are looking at question number 1 on the biology practice air test. This was assigned to you to prepare you for the air test, which is coming up on Tuesday. I'm going to walk through each of these questions. It's going to take several videos, I think, but I'm going to walk through each question and try to give you a little bit of like a tip or a hint about what I would be thinking as I'm trying to answer it. And this hopefully will help you if you get confused or lost anywhere along the way. To help you study and prepare for the kind of things that they're going to be asking of you. So we'll just get started here with number 1. This first question is asking about 4 different cell specimens, and the question is which one is a prokaryote, and to decide that you have 4 sets of characteristics you're looking for here. And the biggest thing for a prokaryote, the first thing I'm going to look for is that prokaryotes don't have organelles and they don't have a nucleus. So the obvious ones here are chloroplasts and DNA in the nucleus. First of all, chloroplasts are organelles, so if it is a yes here, like cell number 1, that means it cannot be the right answer. As far as DNA in the nucleus, this is really the big one. DNA inside the nucleus means there must be a nucleus. So there is no nucleus in a prokaryote. The DNA is floating in the cytoplasm. So DNA in a nucleus, to say no to that means that our DNA, because the DNA's got to be there, the DNA is in the cytoplasm instead. So cell 3 is the correct answer there. By the way, we also have a cell membrane. Yes, every cell has a cell membrane, eukaryotes and prokaryotes. So that doesn't really help us much. As far as a cell wall would be absent if it was an animal cell. Most other cell types have a cell wall. That includes prokaryotes and other eukaryotes like plants. So cell 3, if you look at the characteristics, there's no chloroplast, there's no DNA inside the nucleus. There is DNA, but it's not inside of a nucleus. So that cell 3 is the best answer here. That one's a prokaryote. Next up, question 2. We're looking at, this is about osmosis. Osmosis is the movement of water, and water, just like anything else, goes from a high concentration to a low concentration. So the question here is asking you about proteins and sugars on the outside and inside of the cell, and there's a lot more proteins and sugars out here, and there's a lot less inside. So the concentration gradient for sugars and proteins has a high concentration outside and a low concentration inside. But what that means is on the inside, whatever's left must be filled with water. So there's more water in here, and there's less water out here because there's more of this other stuff. So because there's more water inside, water will move out of this cell. This is a lot like our eggs. This is what happened when we put our egg lab early in the year. We put our eggs inside of Karo syrup, or maybe maple syrup, and the eggs shriveled when they were in the syrup. That's because there was a lot of sugar and other materials outside the egg, and so the water actually moved out of the egg. That's what that egg lab was showing us early this year. Water was moving from where the water was highest to where the water was lowest. So this cell should shrivel up as water leaves. Now you might be thinking that the proteins and sugars should go in, but cells are not permeable to proteins and sugars as nicely as they are to water. Proteins and sugars do not go through the membrane very smoothly. They need help. They need transport proteins and things like that in the membrane. So these particles cannot go through. That means the cell will not swell up because they're moving in. The only option is for the cell to try to balance out the difference here by water moving out of the cell. So the cell will shrink as water moves out. The only answer for that is water goes from the red blood cell into the solution. So that one should be B. Number 3, which structures are present in animal cells and which ones are in bacterial cells? Animal cells, first of all, cell wall. I mentioned that for number 1. Animal cells do not have a cell wall, so there's no check box for that one. DNA, every cell has DNA. That's its genetic material. Animal cells are eukaryotes, just like us, so we have a nucleus in our cells, and every cell needs ribosomes to build their proteins. Ribosomes are the protein builders. That's animal, plant, bacteria, every type of cell. Bacterial cells are prokaryotes, so they do have a cell wall. Bacteria have cell walls, but they do have DNA. That's the next one. The next one is a nucleus, and then again, this is what they are not going to have. They don't have a nucleus. Their DNA is just floating in the cytoplasm. So for them, they also have ribosomes because, again, that's the protein factory, and so those should be our checkboxes. All right, next up. This goes back to transport. This is membrane transport. This question is asking you about diffusion. These particles, something like maybe it's oxygen, tiny particles, usually particles that are not charged, can pass through the membrane. The reason for that is this membrane, if you remember, the little phosphate head parts are on the outside because water is there, and water is polar, and they like polar stuff. Polar and polar, remember, like dissolves like. They like to interact with each other. On the inside, though, those tails are non-polar. They're oily, fatty. Those are the fatty acids. Those tails like to stay away from water. They are non-polar. They like non-polar stuff, so they don't like things that have a charge. So these particles, if they're going to sneak through, this is a snapshot here, before, during, and after. So beforehand, if these particles that are all stacked up on this side are going to diffuse through the membrane, they have to be able to sneak in between these tails. So for them not to have a charge would make life a lot easier. They would be able to sneak through those tails a lot more easily. So for them to move through like this to balance out both sides, again, that's diffusion. We're going down the concentration gradient here. This happens naturally. It does not take any energy. So your job here is to select all the things that apply to diffusion as things go across the membrane. Letter A is true. They probably do not have a charge. Again, non-charged things go through the better because the tails are non-polar. B, it takes energy. No, this is diffusion. Diffusion happens naturally. No energy needed, so no. Again, takes energy from glucose. No, not needed. No energy needed at all. Letter D, it does not require energy. There we go. Letter E, the particles across the membrane could be proteins or carbohydrates. Probably not. We just talked about that with the red blood cell question. Proteins and carbs get stuck on one side. They don't diffuse through the membrane very well. They're too big. A lot of times they have charges, so they don't go through very well. Letter F, the particles crossing the membrane could be water or oxygen. Yes, it could be water or oxygen. Those things diffuse through directly through the membrane because they're small. They're not charged. Water's polar, so it doesn't diffuse as well. Oxygen diffuses really well. Water doesn't diffuse as well, but because it's not charged, it diffuses better than something that would have a charge. So water and oxygen would work there. Letter G, again, strong positive or negative charge, no. So A, D, F for that one. Next up, which organelles give structure and support to animal cells? Letter A, right off the bat, maybe, except cell walls are not present in animal cells, so A doesn't work. B, cytoskeleton and Golgi. Cytoskeleton, yes. Golgi, no. Golgi is for making proteins. Well, actually, it's for transporting proteins. Letter C sounds like a good answer. If we look at D, D does not. Endoplasmic reticulum is also part of the protein shipping process. It is not part of the cell structure. It doesn't help the cell keep its shape. Cell membrane would have been okay, but that's not going to work because endoplasmic reticulum. So again, back to C. That's the one that makes the most sense. The cell membrane provides shape. The cytoskeleton helps. It's like cytoskeleton acts like tent poles inside of a tent. They help to kind of move the outer membrane around and to give the outer membrane some shape. So C works for that one. Alright, next up. This is about a photosynthetic thing and how well it can perform photosynthesis based on temperature. So the graph has temperature here and how much photosynthesis is going on the y-axis. So we see when it's really cold, there's not a lot of photosynthesis. As we get warmer, closer and closer to 25 degrees, we're getting it's really starting to ramp up. We see maximum photosynthesis happening here. And then as we get warmer, it drops off suddenly. Drops off pretty steeply after 25 degrees. So the more warmth we have, the higher the temperature, we get better reactivity here for photosynthesis, but then it hits a limit and then it just kind of drops right off at the end. So we got a couple questions about this. Part A wants to know which statement is supported by the data. The rate of oxygen production is lowest in M. No, oxygen comes from photosynthesis. The higher this is, the higher production we should get. So really B is a good one. The rate of oxygen production is greatest in section M. C, the production of water is greatest in L. No, we're not producing water during photosynthesis. And the number of carbon dioxide molecules in the beaker is greatest in section K. No, that's no. It should be greatest when we have little photosynthesis happening. So B works there. For part B, which statement explains the effect of temperature on the rate of photosynthesis? The biggest thing here, we did this with liver. We reacted liver with peroxide early in the year, and then we treated the liver. We put it in acid, we put it in base, we heated it up, we basically cooked it, and we saw no matter what we did, those things that really changed the liver caused an issue, and we talked about that issue being that the enzymes didn't work properly anymore. And that is the answer to this one. Enzymes involved with photosynthesis get disrupted when we get over 25 degrees, and that is why this goes downhill. Just like that liver didn't want to react with peroxide anymore, the enzymes in the liver were disrupted, and that's what's happening here. The enzymes were disrupted, and that's why it drops off. That's why it doesn't just keep getting steeper and steeper as we go. Alright, we'll stop there and pick up with the next video.